let's start the recording a little bit early so I can just turn it on. Yeah. So is everything's loaded? You're ready to go?
I think so.
Do you have a laser pointer?
Yes.
All right. People are still rolling in. Okay, next we're coming. Hopefully everyone got pizza. So it's a pleasure to introduce Vivian Pera. Vivian, some of you may know her from Photonics West meetings where she's been there, given lots of nice presentations over the years. She's currently a PhD candidate in electrical engineering at Northeastern. She has a great background, so her bachelor's is in physics, where she studied at Harvard. She has a master's from Tufts in electrical engineering as well. And of course, she's a PhD candidate now at Northeastern. That's kind of the regular story, and I won't go into the other parts, but she also has background in literature, in English. She's very well-rounded and articulate and has studied those also at a very, very high level. So hopefully this will be the best articulated seminar we've ever seen. Maybe there'll be a storyline and a plot as well. So let's welcome Vivian.
Oh, goodness. Okay, well, that was quite the introduction. I hope this lives up to the high expectations that have now been set. Thank you for that, Bruce. No, but I'm really just delighted to be out here. Thank you so much for the invitation to speak, and thanks to all of you for taking the time to come. So before I joined Mark Nidre's group at Northeastern, I worked for about 10 years doing statistical signal processing for radar and sonar array applications. I worked at MIT Lincoln Lab. And so when I started working with Mark, it was with an eye towards maybe possibly using some of those techniques to solve some of the image reconstruction problems that we encounter in fluorescence molecular tomography, or FMT. Now, a major difference between the problems that I was working on at Lincoln and problems in FMT is the fact that, the latter are often ill-posed. And so this prevents us from taking advantage of some very well-studied and successful algorithms that are out there. But, what I found is that if I was a little bit creative, in certain cases, I could still avail myself of these techniques. And so what I want to talk to you today is about two algorithms that I'm calling intrinsically regularized, that I came up with for two applications in our lab. And by intrinsically regularized, I mean that the performance doesn't depend on any kind of user-specified parameter. The regularization that is occurring here is being done by the physics of the problem, assumptions that we can all agree on. For example, the fact that optical signals are non-negative. So I'm certainly not claiming that we can do this all the time. But I hope that by sharing with you these two examples that I can maybe inspire you to think a little bit outside the box when you're tackling these kinds of problems. So this is the basic outline of my talk. I know that a lot of you here work on diffuse optical tomography, but I'm just going to very briefly at the beginning go over the basic principles of FMT and say a little bit about regularization. And then I'm going to talk about the two algorithms and the two areas of applications. I'm going to start with the more traditional application, the problem in multiplexed FMT. And then I'm going to tell you about an instrument that I think is kind of unique to our lab, which we call a diffuse fluorescence flow cytometer. And then I'm just going to wrap up with some final thoughts. So, what is fluorescence molecular tomography? Well, it's an optical technique that uses near-infrared light to map the three-dimensional distribution of fluorophores inside a whole animal non-invasively. And in our lab, we work with mice, so that's why there's a picture of a mouse up here. And to talk about fluorescence molecular tomography, I really need to say a little something about fluorophores. The fluorophores that I'm going to talk about here basically are chemical compounds. They absorb light at shorter wavelengths and re-emit it at longer wavelengths. And the important thing to note is that they have a characteristic spectral and temporal response. So they have a signature, in wavelength, and they follow this mono-exponential decay pattern in time. And so the lifetime parameter that's used to characterize them is just simply how long it takes to go from the peak down to the 1/e point. And probably those of you whose work touches, is in very close relationship to the life sciences know that, in recent years there's just been an explosion in the number and variety of fluorescent probes that can provide contrast against specific biomolecular targets. Fluorescence imaging is an incredibly useful thing to be able to do in the life sciences. We can use fluorescent dyes, for example, to stain certain structures. We can modify cells genetically to express fluorescent proteins. So, this can be an entire lecture in and of itself. But, for this talk, suffice it to say that it's a very useful thing to be able to do, and the goal of FMT is to do it in 3D in a whole animal non-invasively. So how do we do this? Well, we do it in a way that's very similar to X-ray CT. So once we've introduced the fluorophore into the animal somehow, we surround it with a series of sources and detectors. And we illuminate the animal with our laser and collect the response at several detectors, such that in the end we wind up with this dense pattern or source detector pair measurements. Now, unlike X-rays, light and near-infrared light propagates diffusely in tissue. It's highly scattering. And so if I ask, where does the light go that enters the tissue at this point and gets detected at this point, I get these banana-shaped volumes that I'm sure you're probably all familiar with. And the fact that this happens degrades our image resolution and means that our image reconstruction problem is what we call an ill-posed problem. So what we typically do next is we come up with this system of equations where on the left-hand side we have our source detector pair measurements. This matrix W is our weight matrix or Jacobian, and that incorporates the details of the light propagation model. And then we have this vectorized image, which is our unknown fluorophore concentration that we're solving for. And the fact that the problem is ill-posed means that in order to invert this matrix, we're going to have to regularize it. And so our solution is going to depend on the assumptions that we use to regularize that problem. However, when we do it, we can get an image out that shows us, where the fluorophore has accumulated in the animal. Now, I mentioned that the problem was, the image reconstruction problem was ill-posed. And in a way, it's similar to this deconvolution problem that I'm illustrating here. We start out with an image of our fluorophore distribution. Our measurement system is, thank you. A measurement system is, limits the information that we're able to acquire about it. And so in the end, we wind up, and of course there's noise too that gets added. So in the end, we wind up with a blurry, noisy version of what we started out with. Now in tomography, of course, what we end up with is something that's not automatically interpretable to us as an image. But we solve these problems in much the same way. We optimize a cost function, and the cost function has two terms. One that is all about matching, making sure that our solution matches our measured data, and the other one that's this regularization term where that encodes some of our assumptions about what we think the solution should look like. For example, maybe we know that it's smooth or sparse. I mean, there are a lot of functions that people can use here. So now, if your assumptions about the solution are correct, then this works. But of course, you still have this regularization parameter lambda that balances these two contributions. And there are theories there about how that should be selected. But I mean, I think ultimately people end up trying a bunch of different values and seeing just kind of what works. And so what I would like to do is kind of get away from this dependency. And so let me now tell you about the first example of that kind of an algorithm. Okay, so I said just a little while ago, I was saying how important fluorescence imaging is. In microscopy, it's pretty much ubiquitous. Images like this are quite common. Here we're using 3 fluorescent proteins to stain different structures in a cell. However, when we image through several centimeters of tissue with FMT, we typically only image 1 or 2 fluorophores at a time. So the goal of this project in our lab is to be able to do multiplexed imaging. So we'd like to be able to image 3 or 4 or even more fluorophores simultaneously with FMT in the same way that they can do it in microscopy. But what are the challenges? Well, since we have to go through several centimeters of tissue, we're limited to this optical window here, which, again, I'm sure is something that you guys are all familiar with. But, this is the part of the spectrum where the absorption coefficient of the major chromophores in tissue reaches a minimum. And so this is a place where we can get, enough signal in and out to take our measurements. And on the right-hand side, I mean, this says 650 to 1.3 microns, but our detectors are not so efficient, really, out beyond 850 or 900. So effectively, this leaves us with about 200 nanometers where we can look between 650 and 850. And here, I've plotted some emission spectra of common organic fluorophore. These are 4 Alexa dyes. And you can see that they're quite broad, first of all, in relationship to the size of the window. And once I start to get like 3 or 4 of them in there, they're really highly overlapped. And the situation is actually even more challenging than that, because since the optical properties are wavelength dependent, we know that the signature that we're going to measure depends on the amount of tissue that the fluorophore emission has had to travel through. So here, what I did was, this is the emission spectra of Alexa Fluor 750, and if my fluorophore is close to the detector, so that it travels very little through the tissue, I get something that looks a lot like what I would measure in a lab, or that you would download from the manufacturer's website. But if it happens to be close to the source, so that it travels through about 2.5 centimeters of tissue, then the shape of this spectrum gets a little bit distorted and shifted towards the longer wavelengths, which is why this is often known as redshifting. And now, if I go ahead and overlay the spectra for Alexa Fluor 790, you can see why this problem is really complicated to solve in the spectral domain, because now my AF 680 looks a lot more like 790 than it does to 680. And so what the solution that we're exploring in our lab is to use both the spectral and temporal information to try to untangle these signals or demix them, as I'm calling the problem. Okay, so to acquire spectral and temporal fluorophore data, we are building an instrument like this. So very briefly, we have this pulsed laser. Our sample sits on this rotation stage here, and we have 3 detectors which are also on a rotation stage. So this allows us to collect light at several different locations, and then we rotate the sample so that we can cover it, over 360 degrees. So each of these detectors is connected to a spectrometer, which is then connected to a multi-anode PMT operated in time result photon counting mode. And so in the end, what we wind up with is this multidimensional data set, that is lifetime information at a number of spectral channels. Now, this data set is quite large, actually. To give you an example, taking into account our spectral and temporal resolution, this represents about 10,000 components per measurement. And we typically employ about 650 measurements to reconstruct a 2-dimensional slice. So that matrix W that I showed you a few slides ago, that would have about 6.5 million rows. So inverting all of this in one shot is really not possible, so we're going to have to do something clever with this. And so the idea that I had was, well, we split the problem up into 2 parts. In the first stage, we do our demixing, so we separate out all our signals, and then we go ahead and we reconstruct each of those fluorophore measurement sets individually in the second stage. And this has several advantages. I mean, first of all, it takes care of that computational complexity issue because we're demixing each source detector pair measurement individually. But it also has this other nice feature too. Now, we know that this problem is ill-posed, and we're going to have to regularize. But this problem, depending on how you pose it, may not be. Or to say it differently, it's less ill-conditioned than this is. So by doing this first and getting it out of the way, we avoid introducing any bias into our demixing that comes from the regularization that we have to do in the tomographic reconstruction. And splitting it up also gives us the benefit that, we can use a number of algorithms in stage 2 here, because out of this stage, we're going to keep all of the information that we measured. So we're not going to downsample here to a number of time gates or a number of spectral channels. We will have that entire wealth of information as if we had measured each fluorophore independently. So what I'm going to do is I'm going to focus on this first stage because that's where the novel contribution is. So to take a look at that a little bit more closely, what we do is we first take this multidimensional data set and we vectorize it. We just concatenate into one long vector. Then we're going to go ahead and employ a linear mixing model. So I'm saying that my measurement is the product of a matrix S, which I'm calling the library, and that contains the known fluorophore signatures of the fluorophores that are potentially in my sample, and this unknown vector of combining coefficients A. And then I'm adding some noise here. And I'm going to say a little bit more about the library in a second, but what I want you to realize is that my library has different sections, one associated with each fluorophore. And likewise, this vector of combining coefficients also can be divided up into those sections. And so what I'm going to do is I'm going to solve for AK using the Lawson and Hansen non-negative least squares algorithm. And once I do that, I recover my underlying fluorophore signatures by taking each section of my combining coefficient vector and multiplying it times the appropriate section of the matrix. And I loop through, I do this for all of my measurements, and in the end, as I said, I have a measurement set for each fluorophore, which I can then go ahead and reconstruct individually in the second stage. Yes?
You already said that the emission spectrum will be dependent on the depth of the tissue.
Yes.
Fluorophore. So in your library, do you take that into consideration?
That is an excellent question, which I am about to address. Okay, so in my library, the goal of my library is basically to include in there a sufficient number of signatures so that I capture the expected variability of the measured signal. So one way to construct a library, and this often is done when people use these approaches, is what I'm going to call the naive library, where if I'm looking for 4 fluorophores, then I have 4 signatures, and this matrix has 4 columns, one for each fluorophore. But as we were just saying, for us, this isn't really a very good solution. So the next level of complexity that I'm going to consider here is if I assume that the optical properties are constant, then I can show that the shape of the signature only depends on the total path length between the source and detector. And the total path length is just simply, the sum of the distance to the source and the distance to the detector. So each of these signatures, one located here, and one here, has a slightly different look. And here I'm showing you 3 examples for Alexa Fluor 680. And you can see that, the difference is not huge. It's just a little bit. But by actually including this variability in the library, we're able to significantly improve our demixing performance. So this library I'm going to call Extended Library 1. And it has 3 signatures per fluorophore. But of course, the more interesting case is the one where we allow the optical properties to vary with wavelength. And here, I need to include a lot more signatures per fluorophore. And you can see why in this plot. So here I'm showing you, this is the spectral and temporal signature for Alexa Fluor 680. The total path length is the same. It's just the relative position of the fluorophore close to the detector, at the midpoint, or close to the source that varies. And you can see that this has a significant effect on the signature, especially where the peaks are in the spectrum. So to adequately capture that variation, as I said, I need to use a lot more signatures. I'm going to use 344 signatures per fluorophore. But if I do that, then I'm good. I can capture that expected variability. Except that something has happened from going from the naive library and Extended Library 1 to this Extended Library 2. And the fact is that those previous 2 matrices were full rank, whereas the matrix that corresponds to Extended Library 2 is rank deficient, because now I have for each fluorophore a number of signatures that are all not that different from one another. So now you might think, well, now if you want to use that non-negative least squares, you're going to have to regularize, which was kind of defeats the purpose. But I'm going to show you that the algorithm as I've described it to you works, even if Extended Library 2 is rank deficient. And I will have a little bit more to say about that later, but first I want to get into how well this process works. So we did some simulations to get an idea of what we could expect from this algorithm. I'm not going to go into all of the details, but if you look here on the upper right, you see that basically I'm assuming that my sample is about 25 millimeters in diameter, which is typical for small animal imaging. And I have a source on the left and 9 detectors in this arrangement on the right, and I'm going to rotate that entire configuration over 360 degrees to get 648 measurements, and each of those measurements has about 7,500 components. And I'm using these Alexa Fluor dyes in this position, and notice that I'm placing the ones that are most similar to one another directly across from each other, which means that they're going to appear often in the same measurements, which tends to make the problem a little bit more challenging. Now we're going to look at 2 cases, one where I assume that the optical properties are constant, and then the other one where I assume that they're wavelength dependent. And these are the values of mu A and mu S prime that I'm utilizing. These are values for skin from this review paper. And so what I do is I generate 10 realizations of each measurement set. Remember, each measurement set has 648 source detector pair measurements. And then I compute this metric, which I'm calling a normalized root mean square error. And what this is this matrix M hat contains the estimates of the Demix fluorophore signatures for, say, the F fluorophore. M contains the true values. This is a Frobenius norm, so it's an RMS difference. And then we just normalize by the magnitude of the original. So this, the way to think about this really, it's a normalized RMS measure. Okay, so these are the results that we get when the optical properties are constant. So we don't expect any redshifting. The red squares represent what we see with the naive library, where we just have the one signature per fluorophore. And the blue circles are what we get with extended library 1, with the 3 signatures per fluorophore. And as I said, there is about a 6 to 10x improvement in demixing performance that we see when we include that extra variability in the library. So that's the performance that we see with this configuration. We also did some trials because, we were thinking what would the effect be of having the fluorophores in different locations. So this blue bar represents the maximum and minimum error that I saw when I did a number of randomized trials where I just dropped the fluorophores in anywhere in the sample. I did 50 of these, and this is what we observed. So, the variation is from about 0.5 to 2%, so not a whole lot of variation. So from this, we take it that performance in this configuration is actually not atypical. Now, the more interesting case, of course, is when the optical properties are allowed to vary with wavelength. And here you see that the naive library approach doesn't work at all, whereas with our extended library, we're able to get really good results on the order of 1 to 2% error. Now, I said I was going to focus on the demixing algorithm, but I just want to show you, if I then take these estimates and I do some tomographic reconstructions, this is why the errors with the naive library are so high, because we're getting a lot of crosstalk. 680 is being misidentified as 700, and to some extent, that's also happening to 750 and 790. But with our extended library 2 approach, that goes away, and we get slightly higher values, which means that we're recovering more accurate concentration estimates. And now these two are CW reconstructions. If we do something where we take into account a number of different time gates, we've published in our group an approach like this before, then we get even sharper reconstructions and even higher values. So it's great that when we do this demixing, we have all of this temporal information still available to us that we can then utilize to get better tomographic reconstructions. All right, so I think I've just convinced you that accounting for this variation in the optical coefficients with wavelength is important, but of course, in order to account for that, you have to know what this is, right? And so we did some simulations to try to figure out what is the impact on our algorithm if we don't get that estimate right. So what I did was I constructed the library assuming this kind of variation for mu A and mu S prime. And then I constructed some signals according to these kinds of variations. And all I've done is basically take that original variation and either exaggerate it or diminish it across my spectral channels. And then I went ahead and I did what I had done before, generated 10 realizations, did the demixing, and this is what we observed. Now, these numbers refer to whether the library is either overestimating or underestimating the variation. And we can see that there's some asymmetry in the results. So it's much better for the library to overestimate the variation. And if you think about it, that kind of makes sense, because that means that if you have less variation than what you were expecting to see, you're still okay. You can still account for the variation you see in your data. But the reverse is not true, of course. But if I go ahead and I do tomographic reconstructions with all of these results, I find that pretty much anything that has an RMS error of less than 10% gives me really good reconstructions, almost indistinguishable by eye. At this level of error, around 20%, we're starting to see a little bit of crosstalk, and then here we're starting to see significant crosstalk, like what I showed you. So, of course, this is just an initial investigation into the issue of model mismatch. We need to do a lot more work on this, but this is encouraging because it tells us that this algorithm doesn't depend on being able to estimate those, coefficients exactly right. Some sort of sense of what the average is and what the, variation looks like may be enough. Okay, so let me get back to this issue of, why does this algorithm work even when S is rank deficient? So here we have our algorithm, just to remind you, we're solving using non-negative least squares, and S is rank deficient, so, this shouldn't work. But we managed to recover the fluorophore signatures like this, and as you can see, we do that really well. So we can understand this by relying on 3 key insights. So the first one is that, typically when people solve this kind of problem, they're really interested in what the coefficients are in that vector A. But for me, I'm only interested in that indirectly, because as soon as I get it, I'm going to do this multiplication, and then, I'll have my underlying fluorophore signature, so I'm essentially going to throw that away. So if my solution is not unique, if there are multiple solutions that give me equally good estimates of my fluorophore signature, that's okay with me. All I need is for my solution to be limited to the correct section of the matrix in order for me to recover the underlying signature like this. So what kind of an algorithm can give us that? Well, there's been some recent work in the computer vision field in an area called sparse subspace clustering. And what these folks have shown is that if you have a matrix that represents a union of subspaces model and you ask for a sparse solution, that limits your solution to the correct subspace. So the way that this applies to our problem is that the library represents a union of subspaces. Each subspace corresponds to a different fluorophore. And then if I ask for a sparse solution in addition to a non-negative solution, I get something that limits my solution to the correct subspace, approximately. And I say approximately because there are little tiny coefficients in other subspaces. This doesn't work perfectly for us like it does in this case, but as you can see, it works really well. So, okay, so we need sparsity and we need non-negativity, right? So we have the non-negativity here, but where's the sparsity coming from? And the surprising thing is that it's actually coming from the Lawson-Hansen non-negative least squares algorithm. Because if you take a look at this code, you see that you can interpret that as a version of non-negative orthogonal matching pursuit. Now, orthogonal matching pursuit is an algorithm that's popular in the compressed sensing community for finding sparse solutions. So it's really interesting that this algorithm that has been around for like 40 years is actually a version of something that started to crop up, maybe 20 years ago. But in my opinion, the way to think about the Lawson-Hansen non-negative least squares is essentially as this multi-purpose tool. If you have an overdetermined problem, it will find that non-negative least square solution for you, just as it has for many years for people. But if you have an underdetermined problem with a sparse solution, it will find a sparse non-negative least square solution. And the beauty of it is that it doesn't require a regularization parameter. And so this is why I say that this algorithm is intrinsically regularized. Okay, so let's see. In the interest of time, I think I'm going to skip over this summary. I think we've pretty much covered everything here. One thing that I did not say. The demixing algorithm, I can demix 650 measurements in about 5 minutes on a laptop with a non-optimized code. So it's very quick. Well, I will say a couple of words about future work for this project. I mean, certainly experimentally validating the algorithm is an important milestone, and that's currently underway in our lab. You know, we have some initial encouraging results, but we still have a ways to go. I also made a number of simplifications with the modeling and the simulation, so I could definitely further develop this model to include more realistic effects. But, you know, I also think that it would be really interesting to do some work in this area and figure out exactly, you know, under what conditions does this thing work with loss and enhance and non-negative least squares? You know, how many subspaces can you have? What are the relationship to each other? How many fluorophores can you accommodate? And, you know, this really hasn't been looked at because this problem is not exactly the same as what the computer vision people are interested in. So there's actually no existing work in this area. And I think it might be very useful because there are other fields that do spectral demixing too. So it could be useful not just for us, but in other areas. Okay, so now let me talk about the second application. And what we're going to do now is we're going to essentially come down by about an order of magnitude. We're going to go from doing FMT over several centimeters to doing FMT over a few millimeters. And the motivation is basically counting rare circulating cells. Now, this is something that, you know, is important in many areas of biomedical research. For example, in studies of the immune system, cancer metastases. But the issue is that the practical operating range of the current methods is limited because they sample very small blood volumes. So this is true when you do peripheral blood sampling. You extract a little bit of blood from the animal. And it's also true of, you know, more recent in vivo methods, like in vivo flow cytometry. And with that, what you do is you're using a modified microscope to look at a small arteriole in the ear or retina of a mouse. But, you know, the blood flow rate there is only 1 to 2 microliters a minute, so it's really an issue if you're interested in detecting rare cells. So the solution that's been developed in our lab is basically to sort of zoom out and look at a larger area, like the limb of a mouse, or the tail of a mouse, where you have larger blood vessels and more blood vessels. And here the flow rate is about 0.2 to half a milliliter per minute. So it's possible to sample the entire blood volume of a mouse in several minutes. But, you know, if all of this is in your field of view, here you can see that we have multiple blood vessels. Here we have an artery, we have 3 veins. Now we have to have some way of localizing where that signal is coming from. Because you don't just want to count cells in your field of view, because you can end up double counting cells as they circulate through the vasculature. So this system was in existence when I joined Mark's lab, and they were using a standard tomographic reconstruction approach to do localization. And so my goal was really to improve the localization performance of this instrument. So let me just briefly tell you a little bit about it. On the left, you see a schematic. We have 2 lasers that illuminate the sample on either side. Then we have a ring of 6 fiber optodes around our sample, and each of these is connected to a high-sensitivity PMT array. We are modulating the sources at 10 hertz out of phase, and so this means that every 0.1 second, we wind up with 12 measurements. So on the right, you see a picture of the instrument with the mouse tail in the ring. Here are the detectors, and here are our 2 sources. And, you know, we've actually used this to collect in vivo data and to validate this concept, and it's been published here and in other papers from our lab. But the data that I'm going to show you today was generated with a phantom like this. So it's a cast polyester resin phantom that's designed to mimic the optical properties of a mouse limb. And then we run this Tygon tubing through it to simulate a large blood vessel. And through that, we use flow cytometry calibration microspheres to mimic fluorescently labeled cells flowing in the animal. Okay, so this is an example of some of the data that we get. Here I'm showing you 6 of the 12 channels. And there's a rather large background that these spikes ride on, which I've removed, and I've just stacked them here so that we can look at them together. And the point that I want to make is that what's going to allow us to tell where the signal is coming from is the fact that you have these spikes that are of varying heights in the different channels. So we can use a standard tomography algorithm, RART, to do some reconstructions. On the left, you see a photograph of the phantom cross-section. This is the Tygon tube, and this inner circle is the inner wall of the tube. And you can see that for the most part, we're doing okay. We can tell where it is in terms of angular position. When the tube is shallow, there's a little bit of bias toward the periphery. But we really run into trouble when the tube is buried deeper in the phantom. However, with this approach, we're not exploiting all of the information that we have. So we're looking for a cell. So we know that the signal is only going to occupy at most 1 pixel. And given our sampling rate, we're taking measurements every 0.1 second. We're looking for a very rare circulating cells. So we can also assume that we're not going to have more than 1 cell in the field of view at the same time. So we can employ a sparsity constraint to improve upon these results. But instead of doing it with a L1 norm, I'm going to use maximum likelihood approach. And the reason that I want to do this is twofold. We know that maximum likelihood algorithms achieve what's called the Cramer-Rao lower bound. And what that is that's a theoretical calculation that tells you for a given data model, this is the absolute best that you could achieve with any algorithm. So, having an algorithm that in theory can do that is really nice. And as you'll see in a minute, this is much quicker than iterative approaches. And of course, you don't need a regularization parameter to do this. Okay, so let's take a little bit of a closer look at my signal model. So I have a measured data vector, which is my 12 measurements. And when I do this, I'm reconstructing each measurement individually, by the way. So I'm only making use of the 12 measurements that I have. Then I have my weight matrix and my vector of unknowns. And I have a background, which I can estimate, and then I'm assuming some channel-independent Poisson noise. So this is my likelihood function. It's basically the probabilistic data model that we have. I'm assuming that I have enough counts so that the Poisson can be approximated with a Gaussian distribution. So I have a Gaussian distribution with this mean and this covariance matrix, which I can estimate from my known background. So when I solve this for the maximum value eta, the solution I get looks like this, which is not very useful to us because this is ill-conditioned at best. And in fact, in the way that I've drawn it, it's actually underdetermined. So a straight application of maximum likelihood for this model doesn't work. However, what I can do is I can reparameterize the problem. So I can split this vector eta up into an unknown amplitude. And by the way, bold for me means either vector or matrix. So this is a scalar. And then this vector E is a vector of all zeros except for a 1 at one location, wherever the cell happens to be. And so now I go ahead and I modify my likelihood function, and now I have 2 unknown parameters to solve for, my scalar eta and the location of this non-zero coefficient. And when I do that, I get solutions that look like this. So this is actually the kind of algorithm that's used in radar and sonar signal processing to solve what's called the direction of arrival problem. So you have a signal that's impinging on your array, and you want to know where it's coming from. And if we look at this expression on the bottom right, it's actually quite simple what's going on there. So the denominator is just a scaling factor. That's a scalar. When we multiply our weight matrix W by this vector E, all we're doing is simply selecting one column of W. And then on the right here, we have our data, which is weighted by the signal-to-noise ratio, essentially. So what this amounts to is you take each column of your weight matrix, you correlate it with your data. And the one that maximizes that correlation is the one that indicates the location of the cell. So if I go here and I plot that function for a particular data vector, I get something that looks like this. Now, this is in log scale, so this peaks looks rather diffuse. But the thing to know about it is that it's smooth and it does have a single peak. I can do some theoretical simulations at various signal-to-noise ratios to determine how well I can localize the position of that peak. And what I see is that in the operating range of our instrument, the location of that peak is unbiased, and I'm able to localize it with better than 50 micron precision in theory. Now, of course, in practice, there are certain things that will degrade that performance, but in theory, it looks like we can do really well with this approach. And then the other thing that I want to mention is that to generate this surface takes about 0.02 seconds. So I had about 2,000 columns in my weight matrix. So this is something that's really fast. So this is something that could potentially be done in real time. And this is an example of the kind of performance that I get from the algorithm. Now, the outer circle corresponds to the circumference of my phantom. The inner circle corresponds to that inner wall of the Tygon tube. Here, these dashed squares are just areas where I've blown them up so that you can basically see the results. Now, these are not tomographic reconstructions. What these are two-dimensional histograms, where the pixels have been color-coded according to how many times the algorithm selected that pixel as the location of the signal. And so you can see that, for the most part, we're doing really well. We can localize the signal when it's coming from a blood vessel that's shallow, and we can also do equally well when the blood vessel is deeper. All in all, we find that, the error from the center of the tube is about 150 to 250 microns, and the spread is about 100 microns, which is well within the diameter of the Tygon tube. These panels on the right, these errors here, which are inflating this value, we actually think that this is due to positioning errors. So in version 1 of the system, we're doing everything by hand. So we try to get these tiny little phantoms in the center of that detection ring, but sometimes we might not be as successful as we think we are. We're just eyeballing it. So in version 2 of the instrument, we're thinking about how to do that better. Okay, so I hope that I've convinced you that when you have a point target model, a really good thing to do is to reparameterize it and to solve it with maximum likelihood. Here are the advantages of maximum likelihood over these other types of algorithms, which I think we've covered. Certainly, it would be nice to be able to extend this approach to situations where we have maybe 2 or 3 cells in the field of view simultaneously. I think you can see what the problem might be. When you have one vector, it's no problem at all to test against all the vectors that you have. If you have 2 possible locations, that becomes very quickly a combinatorial problem. However, we've done some simulations that say if you have some sort of prior anatomical information that can restrict that search space to maybe just where the blood vessels are, then we can maybe get away to using this approach to determine 2 or 3 cells simultaneously in the field of view and still maintain that close to real-time performance. So one thing that I want to say is that, you might be thinking, well, another way to promote sparsity is just to regularize with an L1 norm, right? That's become very popular with the advent of compressed sensing and so forth. This is a situation where L1 doesn't give you the right answer at all. It gives you a sparse solution, but it's just simply not the right sparse solution. So in this case, there's no, readily available convex algorithm that you can just throw at this problem. And then another thing we thought of doing is having, multiple rings so that we can look across time and correlate detections and in that way improve the specificity. So I just want to close with saying that, intrinsically regularized algorithms are certainly possible, not in every case, but for some cases. Sometimes you can get there by reinterpreting a classic algorithm like we did with the Lawson and Hansen non-negative least squares. Sometimes they provide the only viable alternative when the other traditional methods won't work. And of course, the big advantage is that none of these depend on empirically derived or user-dependent parameters. So with that, I just want to acknowledge my advisors at Northeastern, Mark Nidre and Dana Brooks, the other members, student members of our biomedical optics research group, Stacy and Ying, and our funding sources from the NIH, and I'm happy to answer any questions. Thank you.
Thanks, Vivian. So, do we have some questions? Maybe I'll start by getting you warmed up. So, your experiment with the supercontinuum source and the 648 different, wait a minute. Well, lots of measurements. I forgot how many. 648.
Source detector pair measurements, and then each of those has a bunch of different components.
So to add more complexity to that, do you have the potential also for looking at optical properties of both the excitation and the emission wavelengths? Because you've got a tunable source and you've got full spectra that you can access.
Yep.
So have you thought about integrating that in? Because clearly, I think you've shown very nicely that looking at the intrinsic optical property distortions are going to be the difference between accuracy and wildly inaccurate results.
Yeah, so let's see. The simulation component of developing that instrument is significantly ahead of the hardware development part. So Ying Mu is actually the student in our group who's developing the hardware. And I mean, we've certainly thought about that. Haven't really come up with any approaches yet for how we would fold that into the algorithm at this point. But I mean, yeah, it's an excellent point and something that we can do and we should do.
So it also does raise the classic chicken and egg problem, where you need to know the optical properties so that you can account for the optical properties. So do you have some ideas on that? Would that maybe be an iterative approach process, or are iterative things no good?
No, I wouldn't say. I mean, whatever works is good. But I think we've thought about it. It would certainly be great if we could determine this with maybe just a few quick measurements in the lab and then most of the legwork done by some sort of simulation, whether it's Monte Carlo or an analytical model or a hybrid, something like that. But I mean, I think that, we really won't know the answer, unfortunately, until we actually develop. I was talking to someone earlier today about developing phantoms that really realistically mimic the optical properties of tissue in both absorption and scattering. And so we really just have to get in there and figure out exactly, what those variations are and then assess what really is the right thing to do and, how to construct that library.
Thanks.
For those simulations of rank-deficient that you were doing, how many photons do you need if you're doing spectral and light type and mixing?
How many photons do I need?
Yeah, when you're detection.
Well, so I assumed something rather conservative, which was about a 30 dB maximum signal-to-noise ratio. So that, on average, works out to about 18 dB. So that's typical for what we see with the instrument in the lab, with phantoms. So I haven't done too much work in determining, how low could that signal-to-noise ratio go before the whole thing just breaks down. But, I mean, we feel like 30 is kind of reasonable and conservative, so.
I know you guys have some questions.
So if I follow well, actually you were talking about 300 signature, right, for each. Those are the things actually of your temporal profile, or?
So those are the columns in my matrix that correspond to each fluorophore.
So do you obtain them directly from your temporal profile?
Well,
So basically my idea would be like to reduce the complexity of the problem by using the moments of the profile instead of using the bins. So you can use just 3 parameters instead of using 300. Is that feasible?
yeah, so let's see. It might be feasible. I'll tell you, thinking along those lines, so when I initially, came up with that matrix and I realized, oh, it's rank deficient, I said, well, that's no problem because I simply, will just do a transformation and keep only the certain number of vectors that I need to make that, be full rank again. And when I did that, the algorithm didn't work at all. And I think the reason is the following. In order for that to work, I need for things to be non-negative. And when I did that transformation, right now I'm enforcing non-negativity by simply, my matrix is non-negative. And so if I enforce non-negativity on my vector, that's a way of enforcing the non-negativity constraint that the recovered signatures have to be non-negative. That's what I really care about. If my matrix is no longer strictly positive, which is what I would get if I did something like PCA to reduce the dimensionality of that, then I have no easy way of enforcing that the recovered underlying signatures are all strictly positive. And I need that in order to get this property that the solution is limited to the correct subspace. So I'm not sure, if I did what you're suggesting, whether I would still have that property. And I have to find a way to go from those moments then, well, I guess it depends on what my second stage tomographic reconstruction algorithm will look like. If I want to exploit those same moments, then I guess I'm fine. If I want to do something different, then I would have to go from that somehow back to reconstructing the full signature or whatever piece of the signature I need. So.
Yeah, I think a priori, if you stop at the first moment, just the 0 and 1, you avoid the variance, you will not have non-negative problems. That priori.
Yeah. So actually, in real life, the multiple fluorophores kind of mix spatially and spectrally as well. So can you comment on the real case, the multiple fluorophores mix it in the same place? Can you still be able to separate the spectrum?
Yeah, so I think, so there are 2 issues there, right? In simulation, if I put the fluorophores in exactly the same location, the algorithm works just fine. Maybe the error is a little bit higher, but I get really good performance still. In real life, if all of my fluorophores are exactly in the same place or close to one another, et cetera, there may be some effects that I'm not accounting for in this model that, will complicate that. So then the issue would be, can I really, account for those effects in the model in order to make my algorithm work? Because right now I'm not assuming that there's any kind of, interference or going on between the different fluorophores.
I just have an additional comment, actually about the hardware this time.
Yes.
Just behind your spectral meter, you have a multi-channel photomultiplier. I'm just concerned about the crosstalk between the channels, actually.
The crosstalk between the channels.
In the PMT.
Yeah, I'm not the one that's building the instrument. From what I've heard, it's not been an issue so far. But
Those are pretty good designs. It's the basis of the Zeiss microscopes, and so they're pretty robust. So I have another question for you.
yes.
So, there's a lot of mysterious spectra in absorption information content. And sometimes, we have basis spectra of pure components, and we know that the spectra will undergo shifts, and there are unknown components that contribute. So, we may account for 80 or 90% of the composition in the tissue with sort of our known basis spectra of oxyhemoglobin, deoxyhemoglobin, water, and lipid. But we also know that the disposition of those molecules will cause broadening and shifts, and that as tumors grow and develop, there are biochemical pathways that are activated that lead to the breakdown of hemoglobin and the formation of new, unique spectral features within hemoglobin. But we never account for that in the basis. And it seems like, because you're both looking at a library of multiple possibilities, and you also seem to have an approach where you're kind of able to get what is different from the basis out. Have you thought of applying that? Because those things may really, like if you're looking at tumor and breast tissue, they may be a spatially localized property that's quite different from the surrounding.
Yeah, no, that's really interesting. I hadn't considered that particular problem. I think it's all going to boil down to, when I construct that matrix, what can I put in there in terms of these subspaces? How similar can they be? How much overlap can I have before the whole thing breaks down? And I'm not really sure what that answer is, because that really hasn't been studied in a rigorous manner at all.
Yeah.
So all I've done is shown that when I have fluorophores that are very close together in spectrally and also in lifetime, it's still okay. But I mean, who knows where the limits are or what that can accommodate? So I think that would be really interesting to look at.
It's intriguing to think about, because the closeness of the spectra is probably comparable to the closeness of the absorption spectral variations, and those have a lot of underlying meaning from a biochemical and sort of molecular point of view.
Yeah. Yeah, no, it's a good point.
Yeah. Any more questions? Okay, let's thank Vivian again.